Good morning. It's July 4th, and this is your daily brief in science. Here's everything you need to know. A groundbreaking study from the Karolinska Institute in Sweden has revealed the presence of immature neurons in nearly all adult brains examined, except for one. This discovery suggests a potential for adult neurogenesis, a topic that has garnered significant interest in the scientific community. Led by Marta Paterlini and in collaboration with Chalmers University of Technology, the research was funded by various research councils and foundations, underscoring the importance of exploring brain function. The team identified 3 types of neural progenitor cells primarily located in the dentate gyrus of the hippocampus, confirming their presence across different species and enriching them for various markers. The study indicates that all stages of neural progenitor cells are present during early childhood, with proliferating progenitor cells also found in adults. This suggests that neurogenesis may be a continuous process throughout life. The confirmation of ongoing neuron formation in the adult hippocampus was achieved by identifying origin cells similar to those in other mammals, despite some differences in gene expression. These findings could reshape our understanding of lifelong learning, recovery from injuries, and the potential for neural plasticity in adulthood. Interestingly, the research noted that 2 adults had a higher number of neural precursor cells, with one having a history of epilepsy, hinting at a possible connection between neurogenesis and seizure activity. Jonas Friesen, the study leader, emphasized the significance of these findings for understanding human brain function and their implications for regenerative treatments for neurodegenerative and psychiatric disorders. However, skepticism remains among some experts, including Juan Arellano, who pointed out that the progenitor cells were identified using a machine learning algorithm, raising questions about their functional relevance. Despite the identification of these progenitor cells, the study did not quantify their numbers, leading to concerns about whether their presence indicates genuine adult neurogenesis or remnants from embryonic development. While the findings are promising, experts caution that the identified cells may not be true neurons or could be linked to pathological processes rather than healthy neurogenesis. A groundbreaking study from the UC Davis Comprehensive Cancer Center has uncovered a genetic mutation that may clarify why humans are more prone to cancer than chimpanzees. This research, published in Nature Communications, zeroes in on a mutation in the immune protein FA's ligand, where a single amino acid alteration from proline to serine increases its susceptibility to plasmin, an enzyme that tumors use to facilitate metastasis. This mutation highlights an evolutionary trade-off. While it may have supported human brain development, it also appears to heighten cancer risk by weakening immune responses. Joginder Tushar Singh, the senior author of the study, pointed out that this genetic change likely contributed to the growth of human brain size, but compromised our ability to combat cancer effectively, allowing tumors to evade immune detection. FA's ligand plays a crucial role in enabling immune cells to induce programmed cell death in cancer cells. However, the human variant of this protein is structurally less effective compared to that found in chimpanzees and other non-human primates. Elevated levels of plasmin are commonly associated with aggressive solid tumors, such as triple-negative breast cancer and colon cancer, where they undermine treatment effectiveness by neutralizing FA's L. The study also sheds light on why immunotherapies like CAR-T and T-cell therapies show greater success in treating blood cancers, rather than solid tumors that depend on plasmin for metastasis. The researchers suggest that blocking plasmin could potentially restore the cancer-fighting abilities of human immune cells against solid tumors, opening new avenues for treatment. Combining existing cancer therapies with plasmin inhibitors, or antibodies that protect FA's L, may enhance the effectiveness of immunotherapy for solid tumors. Furthermore, research indicates that antibodies targeting FA's ligand can shield it from degradation by plasmin, thereby restoring immune functionality in mouse models, indicating promising therapeutic strategies. These findings emphasize the significance of understanding our evolutionary history in developing new treatment strategies for diseases that predominantly affect modern humans. Overall, this research underscores the critical role of plasmin in aggressive solid tumors and the necessity for further exploration into primate biology to improve cancer immunotherapy outcomes in humans. Researchers at Harvard have unveiled a groundbreaking predictive framework that merges biophysics and artificial intelligence to identify high-risk COVID-19 variants before they escalate into significant public health threats. This innovative approach aims to enhance pandemic preparedness by forecasting viral evolution and enabling timely public health interventions. The first study introduced a model that quantitatively links the biophysical characteristics of the COVID-19 spike protein to the likelihood of a variant becoming dominant. This model addresses limitations of previous predictive methods by incorporating epistasis, recognizing that the effects of mutations can depend on one another, which improves its predictive accuracy. A second study presented the viral framework, which stands for viral identification via rapid active learning. This framework accelerates the identification of high-risk SARS-CoV-2 variants by focusing lab resources on the most concerning mutations. Notably, viral can identify high-risk variants up to 5 times faster than traditional methods and requires less than 1% of the experimental effort, significantly enhancing outbreak response capabilities. The interdisciplinary approach of these studies combines molecular biophysics, artificial intelligence, and virology, and aims to create a proactive biological forecasting platform that can be applied beyond infectious diseases, including challenges in cancer biology. Researchers are also focused on adapting and scaling their framework to address challenges posed by other emerging viruses and rapidly evolving tumor cells. Led by Professor Eugene Shachnovich, the studies have received substantial support from the National Institutes of Health, highlighting the critical role of federal funding in advancing scientific breakthroughs that address major health issues. Shachnovich emphasized that the lab's work aims to better prepare society for new viruses and pandemics by utilizing fundamental principles of physics and chemistry. The research, published in the Proceedings of the National Academy of Sciences, seeks to enhance proactive measures against emerging viruses by forecasting evolutionary changes in viral variants. The interdisciplinary research team includes members from Harvard's Department of Chemistry and Chemical Biology, showcasing collaboration across fields such as molecular biophysics, AI, and virology. Researchers at Hiroshima University have developed a groundbreaking technique to grow mesenchymal stem cells in aligned sheets. This advancement significantly enhances the production of healing proteins essential for tissue repair. The innovative method employs specially designed temperature-responsive culture surfaces with microscopic stripes that guide stem cells to grow in parallel. This alignment mimics the natural arrangement of cells found in muscle and skin tissues. By preserving natural cell-to-cell contact and the extracellular matrix, these aligned sheets enhance the durability and functionality of stem cells compared to individual cells in suspension. The alignment also improves the cooperative functions and communication of stem cells while maintaining their ability to differentiate into other cell types. This advancement holds promise for enhancing targeted regenerative therapies for conditions such as heart disease, liver damage, and autoimmune disorders. Aligned stem cell sheets were found to secrete greater amounts of key healing factors, including vascular endothelial growth factor and hepatocyte growth factor, which are crucial for blood vessel formation and liver repair. The technique also addresses the challenge of individual stem cells dispersing after injection, improving retention at treatment sites through the use of sheet-based delivery. Notably, the method is cost-effective, utilizing simple modifications of existing culture materials, making it scalable for clinical applications in regenerative medicine. The temperature-responsive culture surfaces allow for the gentle harvesting of cell sheets, preserving crucial cell-to-cell connections and the protein matrix. Professor Kenichi Nagase, the study's corresponding author, emphasizes that enhancing the function of mesenchymal stem cell sheets can significantly improve their therapeutic effects in regenerative medicine. The research, conducted by teams from Hiroshima University, Keio University, Tokyo Women's Medical University, and the University of Utah, suggests that these aligned stem cell sheets could lead to more effective and accessible cell-based therapies. The findings of this research are published in the journal Materials Today Bio, highlighting the collaborative efforts of multiple prestigious institutions. Researchers from USC and Johns Hopkins, led by Daniel Lidar, have achieved a groundbreaking milestone in quantum computing by demonstrating an unconditional exponential speedup using IBM's 127-qubit quantum Eagle processors. This breakthrough involved solving a variation of Simon's problem, a significant challenge in quantum computing that illustrates how quantum algorithms can outperform classical ones exponentially. The term unconditional speedup is crucial here, as it indicates that this achievement does not rely on unproven assumptions, setting a new standard for future advancements in the field. The team utilized innovative techniques to achieve this speedup, including limiting data input, compressing quantum logic operations, and employing error mitigation strategies to address noise and decoherence. The research demonstrates that despite the inherent challenges of noise and errors in quantum systems, algorithmic advantages can still be realized, marking a significant step toward practical applications. Lidar emphasized that the quantum computing community is beginning to showcase how current quantum processors can outperform classical systems in specific tasks, highlighting the technology's potential. However, Lidar cautions that practical applications of quantum computing still require further development, particularly in addressing noise and decoherence issues in larger systems. The findings of this study were published in the journal Physical Review X on June 5, 2025, underscoring the significance of this advancement in the quest for practical quantum computing. IBM continues to be a major player in the quantum technology market, investing heavily in the development of large-scale fault-tolerant quantum computers. The market for quantum computing is expanding rapidly, with numerous companies and institutions investing to unlock the technology's full potential. Quantum computing leverages principles of quantum theory to dramatically increase computation speeds, enabling solutions to complex problems that classical computers struggle to handle. This exponential speedup has significant implications for various fields, including computation, drug design, code breaking, and material discovery. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.